Just hold for a second so I can share it. I hope you can see my screen. Can anybody tell me whether this screen is visible there? Okay, so I think everybody's seeing. So this is the Global Institute of Stem Cell Therapy and Research, and I'm also having another company that is Celebration Life Sciences, which is working with the several kind of clinical trials. And so I'm going to discuss today the role of stem cells in neural and hematopoietic disease, and also several other use of the stem cells. And these are the places actually I worked at in Japan and University of California, San Diego Medical School, where I developed this science, in fact. And University of California, Irvine, and Salk Research Institute, and Burnham Institute, San Diego, and UCLA. So this vision of this GeoStar, that is Global Institute of Stem Cell Therapy and Research, is having two divisions, that is therapy division and research division. The research division, where we do research and develop the science, so we can translate this science into the therapy division. And of course, finally, we can serve the humanity. That's the final goal of GeoStar. So I will just show you a little what we do, and this will explain our entire program.
Healing is an absolute necessity, not an option. True healing always begins at the root level. In humans, roots are stem cells. To replace the ineffective conventional treatment methods, to rekindle the lost hopes of suffering patients, to revolutionize the modern age of medical science globally, GeoStar welcomes you to experience the groundbreaking stem cell treatment procedures that now enable us to treat previously incurable diseases like autoimmune diseases, metabolic diseases, and degenerative diseases more efficiently and with almost no side effects, far outperforming traditional medical procedures. Novel cancer cellular immunotherapy has also shown remarkable efficacy for cancer patients, and thanks to the non-invasive nature of the treatment, it's reliable and pain-free.
My name is Dr. Ian Baker, and I became very interested in the work of GeoStar because they represent the new frontier of healing and medical science.
The most colorful, diverse, and happening place in India is the city of Ahmedabad in the western state of Gujarat, which has become a medical tourism destination to begin your healing journey. The city itself will ease your pain and help you heal with its mesmerizing locations, lots of festivals, and welcoming people. When you arrive at the Ahmedabad airport, our representative will already be there to warmly welcome you. The more comfortable your stay is, the more peaceful your healing journey will be. Hence, the luxurious hotels will undoubtedly do their job. Let's visit the talk of the town, the globally recognized ultramodern and most advanced stem cell treatment facility of GeoStar.
So Dr. Divyan Patel, you've seen that with all the other doctors here at this clinic, who have all been guided by Dr. Anand Srivastava in America, and who also comes here often, and the CEO, Devin Patel, who has been so inspiring to me personally in the vision that he has for GeoStar and its future.
So this is just one short introduction what we do. And so I will not really play all the entire video because we have limited time. And so now I'm coming on the main topic. So we know that particularly this is just a representation of a stem cell. It's embryonic stem cell actually. So I'm just making a specific presentation of the stem cell, though we do not use embryonic stem cell in the patient at least right now, but we use for the research purposes. So we know that there is a blastocyst stage and inner cell mass. These inner cell mass are the pluripotent in nature. You can culture this and then you can make hundreds and millions of the cells, and these cells can be changed into any kind of cells, starting from the neural to blood to liver to lung, whatever. You can create entire human basically. So this is the philosophy of stem cell I'm talking. When I started in 2000, 24 years back, at University of California, San Diego Medical School. So entire idea is to take these stem cells, culture it, and then secondly, differentiate towards the different organs, and then you may transplant to the patient, as our previous speaker was telling, and he did wonderful job. In fact, the exosomes combining with mesenchymal stem cells and showed very significant outcome in the liver cirrhosis. So this is the whole concept, taking the stem cells and transplanting to the patient so you can treat any kind of damage of the organs or any kind of degenerative diseases. So let's go to next slide. So basically we have endless possibilities. So I'm coming on very, very beginning, when I was struggling how to culture the stem cell, and that particular time in 2000 or 2001 or 2, we were culturing embryonic stem cells supported by the animal feeder layer of feeder cells. You can see these elongated cells of the feeder fibroblast, which is needed at that particular time. I'm talking 2001 to 3. To support the human embryonic stem cell. But these stem cells you cannot use to human because it is contaminated with animal product. So that was a big challenge in the beginning of the stem cell science, particularly for the regenerative medicine. So I work and with a few people, and then we developed this culture medium, which was not supported by any kind of animal feeder cells. And we got success in creating a culture medium without any kind of contamination. So this is the human embryonic stem cell without any animal feeder. And this certainly can go to the humans for any kind of clinical trial. So taking from this photograph to this photograph, which I'm showing you 2 photographs here only, it took almost 4 years for me to come here from this side to this side. So after this, actually, people started using stem cell for the clinical trial and all these things. It is 2004 or 5, I think. So let's go to the very interesting part of the development of this science. So in very beginning, in fact, nobody was knowing if you inject the stem cell in the body or in vivo, what is going to happen, whether it is going to survive or not. It is going to survive, then it is going to multiply or not, or it is really going to participate in development of any organ or not. So if you see these are the developing embryo, mouse embryo actually. And I was also establishing in 2000 and 2001 or 2 in utero gene therapy. Like when the fetus is developing, and the concept of in utero gene therapy was that, is it possible to change the defective gene to treat any disease or genetic disease when the fetus is developing? So I established that model already in University of California, San Diego Medical School. Using the same model, what I did, I injected the stem cells in developing mouse embryo. And when this embryo started developing, you can see this dark color here, all this body. These are the control. And this is the dark color, which is showing that the stem cells, which I injected, really survived and participating in development of all the organs. This is the data I'm showing from 2003. So this opens the idea that yes, when you inject the stem cell, it really participates in development of all kind of organs. So that was wonderful result and report. So the people may use this stem cell for development or treatment of or repair of damaged organs. So let's go for the next. Now I'm coming on my main point. So I was always thinking, is it possible to really generate the red blood cells or blood cells using the stem cells? Because there are a lot of hematopoietic or blood-related problems, starting from leukemia, lymphoma, sickle cell anemia, thalassemia, lot of autoimmune diseases which are related with the immune system, ultimately it's the blood. And then blood transfusion also is a big problem, because once you need blood, you always has to do three things. First, you have to match the blood. Second, you have to really make sure that this blood is not contaminated, because there you can screen the blood for some kind of contamination, but you can never screen or detect unknown things in the blood which is coming from another or donor. And then third thing, there is never going to be the sufficient supply of the blood, or whenever it is needed you may not get. There is always a scarcity of the blood. To solve all these three problems, I was thinking, why not to try and see if we can generate the red blood cell or blood cells using the stem cells to solve this problem. So then I started differentiating the stem cell towards the red blood cell. And these are the data I'm going to, and this is all published data actually. So anybody can go and get detail, but I'm not going to talk much detail here. Just I'm because of the constraint of the time. So these are the stem cell was differentiated into the embryonic body, and then finally these are the CD34 positive cells. These are the fluorescent cells. So you can see after 14 days, I almost was able to differentiate almost 90% stem cell into the hematopoietic stem cell, that is CD34 positive cells. And these particular CD34 positive cells, which you are seeing here, these cells actually gives all kind of blood cells. So you can make platelets, you can make red blood cell, you can make all whatever you like to make. But to make sure, and there are a lot of other data which I'm not showing here, which can be anybody can get in my publications. So to see that whether this is really the hematopoietic stem cells or not. So the same thing was stained by peroxidase staining, and you can see all the brown things, and that tells that yes, these are the hematopoietic stem cells. But as I told, I was more focused on differentiating these hematopoietic stem cells into the blood cell or RBC, red blood cell. Then I thought, okay, this is the hematopoietic stem cell, so let's try whether we can differentiate these things into the red blood cells or not. So I develop all this specific cytokine-based medium, and then finally I differentiated those hematopoietic stem cells, that is CD34 positive cells, into the pre-erythrocytes. And we know that our red blood cell does not have the nucleus. It's a donut shape. It's without nucleus. But before the maturation, it has big nucleus. So you can see this cells has big nucleus and very less cytoplasm. So, and the size of this is almost 12, 13 micron. So that is the perfect size. But it was a big challenge to remove this nucleus. So in fact, you're telling that some live cells remove your brain, that is a nucleus, but still you have to function. So finally, I developed a medium which could make donut shape red blood cell. This you can say this is reticulocyte. It is about to shed off its nucleus. This is all published. This is Blood and Journal of Translational Medicine, Stem Cell, BBRC. I started working on 2004, 6, 7, 9, 11. So it's a wonderful actually things. And so I published all, but not particularly this part, because we are trying to make the red blood cell in the bioreactor right now. So we are trying to design a bioreactor so we can manufacture large amount of red blood cell. So that's our goal. And so we are under the process. The beauty of this red blood cell is this is O negative. So you can really transfuse these blood cells to anybody. And then second, it is made in controlled environment, that is inside a bioreactor. So the chances of having any kind of disease or contamination is almost zero. And you may have unlimited supply. Whenever it is needed, you can just give it. So that's what we are working right now with the GeoStar. And after the COVID-19, we all know that it's so difficult to get the blood, and blood transfusion is a big challenge after the COVID-19. So this may solve a lot of problems. So, but anyway, we are working right now. So this is our blood story. Now I'm coming to our next side. So that is, then I was studying in the medical school, and medical school was teaching before 2005 that if brain is damaged or neurons are damaged, you may not repair it. Then I thought, is it possible to really repair the damaged neurons using the stem cells? Then again, I developed a mouse model, and what I did, and this is the cartoon representation of my model. So what I did, I inflicted an injury to the one side of the brain, which you can see the red color here. And then I made GFP, green fluorescent stem cells. And that time in 2002 or 3, even making the green fluorescent stem cell was also a big challenge. But I was, as I told, I was working on the gene therapy in 97, 98. And I showed first time, in fact, that in 98, 99, that you may change pancreatic gene with the correct gene, but that is a different story. Here I'm talking stem cells only. So using the same technology, I transfected these stem cells with the GFP and then asked 3 questions. First, when you inject the stem cell, is it going to survive or not? If it is going to survive, it is going to multiply or not? And if it is going to multiply, it is going to repair the damaged portion of brain or not? So these are the 3 questions I asked. A very simple question, because I was not knowing, nobody was knowing that what is going to happen. So when I injected, as I told, I asked 3 things: that surviving, multiplying, or going to repair this damaged portion of brain is not. So let's see the real original data now. So this is the original data. So it was published in, again, the stem cells, 2006. So when I injected the stem cells, fluorescent stem cells, after 24 hours of the injection, I really did not see any fluorescence. In fact, you can see this black color. There's no fluorescence. If you really see under the microscope with the dark room, you may see some green here, very faded green. And you don't know whether it is correct or not. But then to make sure this green is correct or not, then I use the peroxidase staining, and you can see some brown spots here. But again, this is not so much that you can really say anything that it is going to repair or not or whatever. Okay. Then I waited for 1 week. So after 1 week, I see a lot of green, and it's traveling towards the damaged portion of the brain. And this is, again, the superimposed image with the light microscopy. So you can see the blue, and it's traveling towards the damaged portion of the brain. And then this is after 8 weeks, basically. You can see this damage, and these are the stem cells surrounding this damaged portion, trying to repair it. And the most important thing here is that unwanted cells are gone. That is the most important thing, because you do not want unwanted stem cell to present there. So this was a very amazing thing, and I was very excited. Though in beginning I was very, very depressed after seeing the results. Then I did some real-time gene expression and all these things, and this is, again, I told it was published. And this was the first report, in fact, that brain can be repaired using stem cells. So that's the beauty. And then, so this was published. This publication came out after 2005. Then basically the medical school started teaching that if brain is damaged, you may, or neurons are damaged, you may or might repair using stem cells. So seeing this report, actually, this Superman, Christopher Reeve, he fell down from the horse and he broke his spinal cord. And his team also came to know about this, that this publication, that neurons may be repaired using stem cells. And then he really wanted to do this. And then I told it's not possible, in fact, because it's just published and we have to do further research really to know whether we can or anybody can repair the spinal cord injury or not using stem cells. So, but at that particular time, University of California, Irvine has wonderful neurology and spinal cord injury department, and they were working very hard on the spinal cord. So I told, if you may fund or the fund can go there, and so we may try to do this there. And then he funded that. And there's in the UCI, basically, there is Christopher Reeve Irvine Neurology Department, where I joined to work on this clinical trial to repair the spinal cord injury using human embryonic stem cell. And probably I think this is the only clinical trial using embryonic stem cell to repair the spinal cord injury. So again, this is the idea. So as I told, this was the Reeve Irvine Research Center, University of California, Irvine. So we created a mouse model. You can see this is limping. And it was the Hans Christet group, which I was working together. So you can see this signal is going from forebrain to, and then when there is injury, we are on the wheelchair. Okay, and then signal is blocked. So the biggest problem of this spinal cord injury, the most painful situation, mostly the young people get problems because they are biking, doing a lot of sporting things, and they get injury, and all of a sudden their life is totally gone. It's like hell. So you see, then it's damage, and then signal is blocked, and person is paralyzed. So what we did, we used these, as I told, the stem cells, and asked 3 questions, whether it is going to survive or not, multiply or going to repair or not. So when we injected, we see that signal started going, and this mouse started working. You see the tail is up. So that was a wonderful thing. So let me show you. So that was the first clinical trial, and this went finally to Jiran, and then there are a lot of other things, and it never went at the final stage. That was the sad ending of this clinical trial. But anyway, nothing goes in vain, particularly in this science. All information really make a foundation for next stage. So saying that, then I created the neurons using the stem cells in the lab. This is the real neurons. You can see that. Then people can tell, okay, it looks like neurons, but how you know it is neurons? So, as everybody who is sitting here and listening, the scientists, they use specific markers and all these things, so you can make sure that these are the neuronal cells. But again, to know whether it is working or not, so what I did, that I plated the cardiomyocyte in the plate and used this neural cells and plated on top of this. When it infused with this, you can see this cardiomyocyte start beating in the plate. So that tells whatever neurons I made or neural cells, it really works. So that is the best part. So let me go for my next. Okay, so I told you the 2 story that blood making the red blood cell directly, and then neural cells, but immune cells are also the big immune problem, or autoimmune diseases are also the big problem, and that is also somehow related with the blood and defective immune system. So I was thinking, is it possible to really repair the autoimmune disease using the stem cells, and particularly Crohn's disease? And then again, I started from the scratch. I first created the Crohn's model in the lab using the mice. And then once it was established that this mice are expressing the Crohn's disease or having the Crohn's disease, you can see this Crohn's intestine very diffused. Okay, and this is the healthy intestine. So I again did the same thing. I asked 3 questions. When you inject the stem cell, what these cells are going to do? Are these going to repair or not, survive or not? And I was lucky that whatever I did, it worked. So this is again Srivastava and published in 2007. And so let's see. When I injected a stem cell, after 5 days, you can see very 1 or 2 greens, very less cells. Okay, but after 8 weeks, you can see a lot of green, a lot of cells. And then you can see the data here, the normal, here's the Crohn's, and after a stem cell, this came almost the normal level. So that is again the wonderful findings, and probably the first time to show that autoimmune disease like Crohn's maybe or might be repaired using stem cells. And now we know that a lot of people are using mesenchymal stem cell, including us also. And in our different centers, we used mesenchymal stem cell basically to manage the Crohn's disease, and it's showing so wonderful outcomes. So saying that, this is the wonderful report actually. And then we did a lot of things, particularly these. I'm not going to discuss so many things because now we are using a lot of mesenchymal stem cells to repair autoimmune disease and several kind of autoimmune disease. So I'm just showing one Parkinson's, report here using the stem cells in our center. So you see the before, and she has the arthritis and Parkinson's kind of symptoms. And after the treatment, you can see how she is healthy and she can walk even. So this was amazing and very encouraging results. And we are under the process of really applying for the clinical trials for all these things in US and right now. But you can see it's really changing the life of the people. And we are exclusively using mesenchymal stem cell at this time point. So this is the mother talking the same thing. So I'm not going to really. There are a lot of clinical reports and related to the real patient and all these things. I'm not going to show you because it's going to take a lot of time. I think I have few minutes only. And this is again the skin psoriasis. You see, it is published in 19. We just use the exosomes kind of things like and this is 24 and 19. And once we use the conditioned medium, including having exosomes, this psoriasis is gone. You can see this alopecia. This is published in 2021. This is also used by mesenchymal stem cell, and it's gone. So this is wonderful results. And these things, nobody can treat. There's no treatment available. So stem cells really going to change the entire world and the idea of treating the different kind of disease. And this is the most important thing. This is the most natural way, most non-invasive way to treat several diseases. This is the arthritis, the real arthritis patient data. You can see this came to the normal level. You can see this RF factor and this is C-reactive protein actually. So you can see this went up and after the treatment it came down. You can see the rheumatoid RF factor. It was 23 in June 4th, and in November it is 11 in 3-4 months. So this is wonderful reports of different kind of autoimmune disease. We have a lot of reports. We have treated more than 20,000 patients. So same is going with the diabetes. And we are under the process of US FDA clinical trial. We just waiting for our approval to treat the type 2 diabetes. So there are a lot of things you can use for the coronary artery disease and all these things. And this is the diabetes data. When you use the stem cell, blood sugar level comes to the normal almost. And this is coronary artery. I'm not going to really a lot of things we are doing, and particularly when this is the most important actually story which I would like to really share, and after that I will conclude my talk. When the COVID-19 came in early 2020, there's no treatment, and thousands of people were dying. So I was thinking, is it possible for me to really manage somehow? Because no treatment is there. And I immediately wrote a protocol in February 2020 and then sent to FDA. FDA approved our protocol in just 2 weeks. And for the emergency use of the stem cell to manage the COVID-19 patient. And these are the just mechanism and all these things, how it works and all this, how a stem cell work in the COVID-19. So we treated the patient. You can see there's a lot of lung and problem and then it was cleared almost. So we saved the patient using the stem cells who was under coma almost for 8 weeks. And we infused the mesenchymal stem cells and we brought back that patient. So that was the wonderful report, and that was came on the news all over the US.
Our ABC attendance reporter John Horn explains why the new treatment could be game-changing and while we await a vaccine.
He was having a stroke. He was under dialysis. Liver function was declining.
The prognosis grim for this 53-year-old COVID patient deteriorating in a New Jersey hospital. The man fell into a coma that would last nearly 60 days.
Nothing was working, in fact, like plasma therapy and all the classical kind of treatment.
That's when San Diego doctor Anand Srivastava and his team at GeoStar sprung into action. GeoStar is an international stem cell research firm based in the Golden Triangle. It got special clearance from the FDA to infuse the 53-year-old man with mesenchymal stem cells taken from an umbilical cord.
Slowly, his renal and liver function came towards the normal. He came out from the intubation, and now he's talking.
Srivastava says it took about 3 weeks for the man to recover. GeoStar interviewed the patient's family, keeping identities confidential due to privacy laws.
Basically, we had no hopes for him, but it turned around.
I feel so happy to say, because he has a daughter and wife, of course.
Srivastava says this kind of treatment could be key in helping COVID patients survive until a vaccine arrives to protect people from infection. John Horn, ABC 10 News. GeoStar is planning to conduct a double-blind study for the treatment to make sure that it's...
So this is all, there are a lot of things we are doing, which I'm not going to because of time. And we are meeting several people, topmost people of the world. So these are the places where I work. I'm going fast now because really I do not want to bore with these kind of things. These are our hospitals, several places, several people. Let me go fast. Finally, these are our hospitals all over the world. This is very important slide because I struggle a lot from working in the field of stem cells, and particularly with the beginning in 2000 to 2004-5. So this is the inspiring sentence for me, that many people, specifically ignorant people, want to punish you for speaking truth, for being correct, for being you. Never apologize for being correct or for being years ahead of your time. If you are right and you know it, speak your mind. Even if you are a minority of one, the truth is still the truth. So thank you very much. And that's all I wanted to talk. And if I have taken extra time, I'm sorry. So that's all. And again, thank you for giving me opportunity to speak in this extraordinary conference.